This is a production of Cornell University.
So it's fantastic to be back in this room. Thank you all so much for coming. Everyone can read, so that I don't need to read the title of my talk, but I'd love to just tell you about a lot of different hemp work that's happening in Geneva right now. All right, so let me just begin with a little bit of intention. Here's an overview, a little bit of background about me and hemp in general. I'd like to talk about the status of the germplasm collection in Geneva. And then kind of three topics. How do we phenotype hemp? What kind of data are we collecting? What does the range and distribution of that data look like? Our lab places a pretty big emphasis on developing tools and methods as we're a public service agency. What are some of the long-term projects we can push on that will provide a lot of value to everyone else long-term? That's really fun and exciting to me to be able to do that and tell you about some of that work. And then finally, genotyping efforts on hemp. So all about me. So 200 years ago, I mean, in 2020, I was a graduate student. I did my A exam in this very room. So I'm, dealing with a little bit of trauma. But I'll get through it. So I had such a wonderful opportunity to learn from Thomas Bjorkman and his deep and wide knowledge and wisdom. We had so much fun exploring the genetic diversity of broccoli together. Broccoli slide in a hemp talk. This is some of the work that we did, some of the varieties we looked at, and the population structures on the side. This had the dubious distinction of going viral on Reddit. So I woke up with like 14,000 notifications on my phone one time. Since then, I have become a curator and a geneticist at the USDA Plant Genetic Resources Unit in Geneva. I love this work. It's so exciting, and it feels like a continuation of a lot of the work and knowledge that I gained here at Cornell. And then there's so many things to talk about today. I'm going to do my best to cover them. And I really want to stress that nearly everything I'm going to discuss today is collaborative. I'll do my best to give everybody credit for the contributions that they've made. And please know, in many cases, I'm just supporting their work. So what is hemp? What is hemp? We like to put a cheeky emphasis on is. So, it's actually kind of a more subtle question than it might immediately seem. So there's a legal definition of hemp. So below 0.3% total THC. I know it's always a great way to engage people to put a biochemical pathway slide on a presentation. And I'm doing this because I like to torture audiences, but I think this is important. So broadly, we can talk about this quote unquote B locus with two alleles. I'm going to oversimplify this a bit, but our two most famous cannabinoids, CBD and THC, are decarboxylated from an acidic form, and those two acidic forms are converted from a common molecule which they compete for, CBGA. So here's some data where we've looked at compared total THC versus total THC, and you find this pretty dramatic pattern when you look at a lot of diverse germplasm. So we talk about these three chemotypes. You can have the BT allele, which is THC dominant, mostly THC. You can have two copies of the other allele, and those are CBD dominant, and then you can be heterozygous for both of those alleles. And that has very profound implications on the types of cannabinoids that you'll observe in a given individual. And here's kind of the critical point is that the legal threshold is set at 0.3% total THC. So note that all chemotypes can run hot. Below that line is hemp, above that line is marijuana, as far as our current laws and regulations are concerned. All right, dioecious. So within hemp, you have distinct male individuals and female individuals, individuals with pistillate flowers and staminate flowers, except when it's not. So that's kind of the bonkers but exciting thing about hemp is there always seems to be an exception. So we have these two pictures that I took about a week ago of two individuals growing in a field trial. On the left is a female inflorescence with anthers protruding out of it. And on the right is an individual with both female and male inflorescences. What's going on here? There's a lot to be said about this, and it's going to take many people smarter than me to disentangle this question. Hemp is wind pollinated. So many of the crops we curate in Geneva are insect pollinated. So think about all these different vegetables. And that's a relatively straightforward process to create pure seeds from those lines. You grow them, transplant them into a field, put an insect exclusion cage over the top, introduce a pollinator, allow random mating to occur, come back and collect the seeds. This is just not possible with hemp. So hemp pollen is about 22 microns in diameter, and depending on who you ask, it can travel 10 kilometers by wind. There's some debate about that, but the take-home message is we cannot trust outdoor regenerations of given accessions of hemp. So we have to use greenhouses and growth chambers and expensive and harder techniques. So hemp is considered a short day where the length of the night regulates the transition from vegetative state to reproductive state. Any guesses what the next thing is? Except when it's not, right? So there's almost certainly more to the story, right? There's kind of some wonderful work kind of mapping the genetic control of that photoperiod sensitivity. But that's not the complete story. There's temperature and cultural conditions and stress that are all cross-talking in there. To my knowledge, this has not been disentangled. There's also photoperiod insensitive varieties that just flower immediately. Multi-purpose. So this is a wonderful slide in the Cornell Hemp Handbook. And to my knowledge, there's no crop that you can produce more different kinds of products from, or it's one of the highest. So depending on the organs of the plant, you can make all kinds of things from construction material to fibers, geotextiles. You can use the wood, the hurd to make construction materials. The seeds can be very rich and abundant and different fatty acids, omega-3s, et cetera, that can be used in industrial processes or food. And then, flowers. People love this plant and it has medicinal and recreational properties, right? So all of these things are part of the biology and part of how humans utilize this plant. This is not even a comprehensive list. This is another point that I want to kind of impress on everyone that I collected Latin long data from different herbaria collections, iNaturalist, GBIF, other collections over time, and plotted this data out. And so cannabis has a cosmopolitan distribution. It's globally distributed. So in these different environments, it's undergone artificial selection by people that are making selections for different purposes. And then in many places, hemp or cannabis has become naturalized, and it's undergone natural selection in these different environments. So there's a proliferation of genetic diversity in this one single species. Okay, so what's the story with the hemp germplasm collection? So we're part of the national plant germplasm system. There's approximately 20 gene banks across the country, and we maintain about 600,000 active accessions that a breeder or researcher can request today via an online portal called GRIN-Global. In Geneva, we maintain a lot of really fantastic questions. So we have the largest apple collection in the world, outright, full stop. We maintain cold hardy grapes, 13,000 unique vegetable accessions across about 12 or 13 genera, depending on how you count it. Hemp was added to this list, and we really began this work in 2020 in full. Hemp has been around in the previous iteration of NPGS, the plant introduction system. There had received materials. This is likely from the Vavilov collection back in the day. So the PI numbers count up sequentially, and they're in the 600,000s now. This is PI 948. This collection is lost, and it's probably priceless, but let's continue. So what are our objectives here in Geneva? So first and foremost is to try to do our best to capture all of this genetic diversity. There's accessions from all over the world, like I mentioned, with all different selective pressures that have acted upon them. So capturing seeds from these different accessions, storing them under optimal seed storage conditions, backing them up in secondary or tertiary gene banks. Some of the seeds we make in Geneva will find their way to the Svalbard seed vault in Norway, for example. We distribute regulatory compliant germplasm to an international community at no cost to provide unique alleles diversity to breeders, to help them develop locally adapted and climate change resistant cultivars. I manage a database which populates a website that's full of phenotypic and genotypic information that the public, mostly breeders and researchers, can access and use that to help empower their research. And finally, we develop data, tools, and methods that we believe will provide long-term value to the broader research community. Some of the traits that we're focusing on right now are plant architecture, which has pretty profound implications on breeding and ultimately agronomic productivity. Flowering time is very important. So as a plant transitions from a vegetative state to a reproductive state, it doesn't really grow much more. It diverts its energy into making seeds. So later flowering varieties tend to make greater biomass and higher yields. So understanding this flowering trait is important. Secondary metabolites. So this is absolutely essential to identify accessions that are regulatory compliant, that stay consistently below this 0.3% THC. Also, identifying unique cannabinoids. We still don't know everything, and these gentlemen, for example, are working on that question. Seeds, fatty acids, proteins. What's the total amount? What's the profile? We're lucky to work with some other USDA labs that are looking into this question. There's a USDA group in New Orleans and Mississippi that are thinking a lot about variation in fiber quality in the stems of hemp. and we're providing them all kinds of materials. So identifying an accession with really fantastic fiber quality and really awful fiber quality, cross them, make an F2 population, and look how the progeny segregate. And you can identify genes that control things like fiber fineness. So here's a infographic of the accessions in our collection right now. It's about up to 700. And many of these come from Central, Eastern, Southeastern Asia. Broadly, there's debate, but Central Asia is the centers of domestication, and following this Pavlovian model, probably where the most genetic diversity occurs. So we place a very high emphasis on land races from Nepal, northern Pakistan, Tibet, those kinds of regions. Here's a kind of distribution about the germplasm by geography. This is how the collection has grown over time. So like I said, we really began adding materials at the beginning of 2022, and it's growing pretty rapidly and consistently. I need to update this graph actually, because it's probably going to be a little over 700 now. And then these are the seed samples that we've distributed to different breeders and researchers. And this has been pretty consistent growth. There's what we believe a pretty strong backed up demand for diverse hemp germplasm. I am legally required to talk about GRIN-Global and give you a crash course. I don't know if you've ever had a librarian come to your class and be like, use the library, it's more than books. There's kind of a similar effect here. So here I've gone to GRIN-Global, that QR code takes you to this website, and I'm running a search here for cannabis sativa. You can also look for unique accessions. So here's New York Feral. This was the first accession added to our collection. It was donated by the Smart Lab at Cornell. And we can see this accession is available because it has the blue shopping cart. Click that cart, order it, seeds will find their way to you. We also collect a lot of metadata or passport information as well. Every accession has their own web page that aggregates all of this information together. So this accession was collected on what was described as a colonial era hemp farm / rope factory. And evidently it seems like that population has persisted over time. So really important, and that provides some context on things like heterozygosity and local adaptation. And then we have oodles of phenotypic data that's also linked to that accession. So things like cannabinoids and flowering time can all be captured and displayed via searching. All right, so let's talk about phenotyping efforts. This is a kind of a schematic of how these phenotypes are collected by different collaborators. Oftentimes we'll send seeds to the collaborator and they'll run an assay or a field trial and return data to us, and I will upload it into the database, and then breeders and researchers can access that data. This is a grant that we got with a group led by Charlie Brummer and Daniel Papp at UC Davis. And we've been growing the same germplasm in these 6 different environments and looking at a giant swath of different traits. So flowering time and sex, architecture, collecting samples for fiber analysis, collecting seed samples for fatty acids and proteins. And a trial like this allows us to begin to pull apart genotype by environmental effects, right? So is one accession really high in this environment, but low in this environment? Those kinds of questions are probably really important. This is the trial that we ran this year. Multiple times a week, every plant got phenotyped for multiple traits. This has been honestly a backbreaking effort, but we're going to get an enormous amount of data out of it. So there's about 500 accessions that are replicated, and we're looking at all of those things I described. This was, you can see the car in the bottom corner just for scale, and this is before the canopy closed, so it's a jungle in there. Believe it or not, you know, back end of October, there's still accessions that haven't flowered yet, which is wild. Our lab publishes this phenotyping handbook that covers in gory details. There's pictures and equations, and it's not a good bedtime read, but it has a lot of info about these different phenotypes that we look at. Not trying to tell people how to phenotype hemp. It's trying to show our work, right? How do we get this data? Folks are welcome to emulate that or come up with a better plan, but we think that it's really important to be hyper transparent with these research efforts. So speaking of plant architecture, on the right, we have 3 plants from one accession and 3 from another. And that's just very dramatic different morphology and architecture, right? So the ones on the right are a fiber variety, and you want tall, thin plants with high apical dominance and relatively low branching. And on the left is a CBD type, where you want more branching, lower apical dominance, more flower production. So how can we best capture this kind of data and make it available to folks? And then on the far left, we see this is kind of our favorite accession because it's so cute and weird, and it's approximately 1 Usoon unit tall.
Zach,
Yes, sir.
I wonder, does the sex of the plant matter for fiber?
Yes, it does. And there's a lot of different theories and thoughts, and
Except when it doesn't.
and a big bottleneck is we don't have a firm grip on what fiber quality is. So it's hard to make concrete statements about it, but there's a lot of beliefs, you know, the males are better, so forth and so on.
Okay.
So we can visualize all of this data here using this kite model that was developed at Cornell. And it's a really elegant way to look at a lot of data at once and kind of gain an overview. So if you can see, all the black lines are an observation of an individual plant, and the colored polygon is the mean. We can look at variation in flowering time. So this is days from sowing to female terminal flowering, and there's quite a range. And this isn't even, I chose this graph because it was small enough to fit on the slide. So it's kind of a lot of big data that we're trying to turn over to support people that are working on these kinds of questions. Very lucky to work with many different chemists. Historically, we've worked a lot with Corey Brownstein, who has he kind of describes himself as a ethnobotanist / analytic chemist. Really cool background, and he's been able to just grind through thousands of samples and return that data to us. So we can look at this and kind of gain an idea about chemical profiles. We're working with these gentlemen to kind of go deeper and look for more obscure boutique cannabinoids as well. We also have a collaboration with Roque, who's a oil chemist, and he's been looking at seed samples and composition of these different fatty acids. We can compare between these different environments. So this is data from UC Davis in Geneva, New York. And I was actually kind of surprised at the R squareds, you know, are in the 0.8 or 0.9 range. It depends on the trait, so forth and so on. But there's likely fairly high heritability to a lot of these traits. We've had a great time this year working with the Chris and Larry Smart Labs. So we've been screening germplasm in our collection for several different diseases, including powdery mildew and septoria. Yes, sir.
How do you protect your fields from the local teenagers?
That's the worst pest in the world. Yeah, so we have a 8-foot high fence and security cameras that I can monitor on my phone, and a very like aggressively friendly stray cat that hangs out in there too. But that's a good question. And some of these plots have not our plots, but this is known to happen. So Lou and Jocelyn evaluated all of this different germplasm in this replicated trial, and had found some pretty promising results. So along the x-axis are these different accessions in our collection. Taylor's done a lot of work with this too. And we see quite a range, and there's some germplasm that looks really promising. Same with powdery. So, these lines may not be particularly good cultivars, but you could use them in a back crossing scheme to move those alleles into your more elite lines. Some tool and method developments. I'm a little behind schedule already. So the idea about regenerating all of this germplasm and maintaining purity, not allowing cross-pollination, is a tough one, and we must increase our throughput. So we're trying a lot of these different designs to do regenerations in a field. So we've made several iterations of these hemp huts that are based on special fabric with 15 micron pore size, which we know should be competent to exclude pollen. They're collapsible, modular. We can fit them over a directly seeded plot. We run negative controls in these, so feminized or female-only populations are grown in some of the tents, and we crack them open if we find seeds we know they don't work, right? So we're still in testing and design, and it's a lot of scientists trying to build things, and so it may not be the most elegant, but we think this is really important. And the goal is to publish the blueprint, make it publicly available, and help other folks. This has been one of my favorite parts of this job, is to work with different groups, especially Breeding Insights, who's based right here, which is a USDA-funded Cornell project. And these guys are, wizards. So we were interested in quantifying leaf morphology. And this gentleman, AJ, cooked up a tool from scratch that relies on this imaging template with the colored dots, which are known diameter and distance from each other. You can take a picture with a cell phone that has the Fieldbook app on it, which tags that image with the appropriate metadata. Then you can his pipeline does some fancy linear algebra and rotates that image and crops it. And then you can extract out all of this kind of data. So things like area and width, morphology, but also you can count every single serration on the leaf, which is wild to me. And so instead of trying to cut leaves and put them in Ziploc bags and driving to the lab and image them under a hot lamp and they're wilting, just do it in the field. The throughput is orders of magnitude higher. It can even handle multiple images and separate them. So incredible work. AJ has even been able to kind of use this data to pull out G/E effects, so same germplasm, different environments, and seeing kind of broader patterns. We think pollen is going to be absolutely essential to the long-term success of our project. So distributing seeds and pollen in parallel. So it allows breeders to get a lot more alleles quickly to make crosses. Pollen is also great because it can break the temporal separation in flowering time. So when one female parent is ready, you just pull the pollen out and you don't have to try to coordinate that with the male flower. This is really hard work, and to the best of our knowledge, there's no really good protocols or standard operating procedures on how to do this. Pollen viability drops very quickly. So this is some of the work that Gabi has been running. And by default, by 12 hours, you have less than 5% or so viability. So you probably could put pollen in a freezer and hold on to it for several months and make a cross. But as we're a gene bank, we need to push that envelope to its absolute extreme. So we're thinking, can we hold on to viable pollen for 10 years? So we're kind of moving through this very complicated search space of different parameters that will enable long-term pollen storage. We made this contraption, which is a spore trap connected to a backpack vacuum that allows us to hoover up oodles of pollen. So Dan is collecting like 10 grams of pollen from a single plant in under a minute, which is incredible to me. And then, we're germinating the pollen under BK media here. The media kind of picked up on this because we published the template and how we made it. So I think I need to move a little faster. Sorry, Gabi. So this question of hemp fiber quality is a really big one. It's hard for people to get their heads around, and it's complicated and probably controlled by a lot of different genes. We're working with this USDA group that has historically done a lot of the cotton fiber quality standards. And so hemp is very different, but they're trying to figure out what tools they can adapt versus tools they need to make. Here's kind of some variation in the types of fiber products. This is an image on the right that Tony made, 4 cultivars on the rows, and the columns are 0 to 4, 6, and 8 weeks of retting. So this allows the fibers to be more readily separated from the stems. So there's a genetic component to it as well as very important agronomic and cultural conditions. So this group that we're working with developed all of this equipment. They made this lab-grade decorticator from scratch. So you can feed a single stem through here, pulls the fiber out, measures the energy required to remove the fiber from that stem, collects other data, and then that fiber gets pushed into all these other types of equipment. So tensile strength, optical sensing, color, you name it. Oh no, sorry about this. So we can look at many different traits that we think will likely be linked to this concept of fiber quality. Work a lot with my friend and colleague, Tyler Gordon, who is kind of trying to take the germplasm to the next level. So he's doing a lot of pre-breeding and early selection and selfing. So we coordinate pretty well during the selfing process, where we'll run those chemotype markers on it, and he can select out regulatory compliant materials and begin selfing them down. Then we can distribute that seed from a otherwise non-compliant accession to breeders and researchers. Finally, and overwhelmingly, we have this enormous genotyping sequencing project that we've done this year. This is in collaboration with our friends at the Hudson Alpha Institute, Alex Harkis and Phil Bentz. So we have collected approximately 4 individuals from 500 accessions for PacBio hi-fi low pass sequencing. They are particularly interested in sex expression in cannabis. good luck. They'll figure it out, I'm sure. But as a germplasm curator, I want to know the population structure, the diversity of this giant pool of germplasm from all over the place. And I believe that this will guide a lot of breeding and conservation and population development efforts downstream. I think the future of germplasm collections will be, you can order seeds and download genomic data or download a genome all at once. I think that's really exciting and just now becoming plausible. And we're excited about developing different mapping populations. We've seen this slide, but this year we've found 2,000 individuals from this field, and we've had to use molecular markers to track down males and females to send tissue on dry ice down to Alabama. And then finally, we're working with Breeding Insight, again, to develop this public marker panel system. So the idea is, can we take advantage of this germplasm collection to make a marker panel that's very aware of all these different kinds of hemp, but it needs to be reliable and low cost. And then the additional thing is we want this to be community driven. So we're able to choose about 3,000 sites. And we've asked many different stakeholders, what's a SNP of interest? What do you use in your group? And many of those folks have already contributed different genetic markers that we can include in this panel. So the goal is to make a community resource that will benefit everyone. George Stack is leading this effort. It's been great to have him on our team. And Danyan is the genomics coordinator at Breeding Insights, and she's a delight to work with and has been giving us a lot of guidance on this process as well. In fact, here's Danyan's slide with some really nice graphics. And this shows how a breeder might take advantage of this mid-coverage genotyping platform. This was a hard slide to make to cram people on here. I know I've missed folks. The best part of this job is the people and working with so many brilliant and interesting folks. I think we need a space race for hemp. The research is 50 to 70 years behind, and we need a lot of resources and people kind of addressing different topics. I love to be a germplasm curator because we're kind of in the middle of that. We can look at a lot of these questions through the lens of genetic diversity and find variation that occurs in here. I also really want to thank germplasm donors, folks from all over the world that have stewarded and maintained these crops. Hasn't always been an easy thing to do. And I'm very appreciative of their willingness to contribute some of those materials to this public germplasm collection. So I think we can leave it here. Questions, I will try to repeat them and also ask Geneva if they have any questions. So let's, I guess, start with Geneva, or pardon me, Ithaca folks.
Questions here?
Have you thought about the impact on your program if high THC cannabis is legalized or allowed? You can't work on it at the federal level now, but any implication that would expand, double your program or?
Yeah, that's a great question. It's hypothetical, and that's one for the politicians and the voters to figure out. And I don't want to speculate too much on what that future scenario might look like. That being said, again, people use this species for many different purposes, and they have different sets of priorities. So is that conceivable? I can't rule it out yet. And in fact, we do hold a DEA registration. So we can, it's kind of a chicken and egg thing where we can't distribute it until we know it has less than 0.3% total THC, but we don't know that till we grow it and test it, so forth and so on. So that's why we have that protection or authorization from the DEA. But it's hard to say. I don't know. But a good question. Next. Yes, ma'am.
So in terms of you're maintaining a lot of accessions, what does that maintenance look like in terms of how often do you have to, pull seeds out, grow them, collect more seeds just to keep it comfortably maintained?
Great question. And that's super important. And I lay in bed and think about this a lot, right? So first and foremost, as hemp has been outside of the scientific world for so long, we don't have a lot of longitudinal long-term studies on how long seed viability can be maintained for under optimal conditions. I can tell you that I've germinated some seeds that were made in the 90s, which is encouraging. The discouraging fact is there's kind of an inverse relationship between the oil content of seeds and longevity, broadly speaking, and hemp is generally considered high oil. We don't know what the maximum lifespan you can juice out of hemp seeds. I can tell you that we're making a lot of efforts to scale up regenerations, and the growth chambers and greenhouses that we have access to, we're running 24/7 right now. And then we're also very lucky to work with many different collaborators, including some of the SUNY system. For example, Morrisville has a large growth chamber, and she's working, Kelly Hennigan is working to regenerate a lot of these materials for us as well. So it's going to take a village. There's some other systems where we can use small exclusion tents inside of growth chambers to kind of multiply the number of accessions that can be regenerated at once. But it's hard to do it right.
Thank you.
You bet. Yes.
So you have mentioned that phenotyping is really a hard work, like backbreakers. Do you consider about using these UAVs for collecting different data sets in the future? Or?
Yes, great question. There's a lot of work at Cornell that are applying UAVs to this. Malin Murdoch is kind of a genius. She's in the SMART lab and has been doing a lot of work to train different models. Can you remote sense for cannabinoids, for example? Maybe. I haven't seen some of her latest data. There's also work being done with robotics, driving a robot through imaging, making 3D images of the plants, that kind of stuff. I think this is probably broadly true for agricultural science, that we'll see a greater amount of integration of these kinds of high throughput phenotyping tools like drones and robots. And I'm very excited to apply those. But there's also a lot to be said for good old yardstick horticulture as well. Get out there, look at a plant and measure it. Okay, Geneva questions. So are you or anyone in your group looking to identify herbicide tolerant traits from this genetically diverse germplasm? Yeah, a little bit. So this would be a great question for Lynn Sosnowski. We haven't run a herbicide resistance trial yet, but I think that's a good idea. And that might be something we could tag team with Lynn on. Okay, I think that might be, Sean says, do monoecious individuals self-fertilize? I think that is quite possible. I'm not going to say in every instance that will occur. I'm not aware of like self-incompatibility work in hemp per se, but broadly speaking, monoecious individuals are considered possible to self-fertilize. All right, I think that's all of our questions.
Okay.
All right, thanks everyone.
This has been a production of Cornell University, on the web at cornell.edu.